It's now my pleasure to welcome Professor George Mashour again to the podium. This is the second plenary talk that George has given. I've already heard a wonderful lecture on consciousness, anaesthesia and cortical communication, which was most enjoyable the other day. Professor Mashour, for those of you that weren't here, received his MD and PhD from Georgetown University and was awarded a Fulbright Scholarship for neuroscience research, which was conducted in both Berlin and Bonn in Germany. He trained in anaesthesiology at Massachusetts General Hospital and Harvard Medical School and had a fellowship and training in neuroanaesthesia at the University of Michigan. He is now the Associate Chair for Faculty Affairs in the Department of Anaesthesiology at the University of Michigan, and his faculty appointments in anaesthesiology, neurosurgery and neuroscience. His primary scholarly interest is in consciousness and anaesthesia. George has been a prolific publisher with over 100 publications and involvement as editor in 4 textbooks related to anaesthesiology and neurosciences. It's my great pleasure now to introduce Professor George Mashour to deliver his lecture, Interfaces of Sleep and Anaesthesia.
Thank you very much for the kind introduction. Again, I'd like to thank the organizers for the invitation to speak here and the sponsors, Covidian, who helped facilitate my travel. So this morning I'm going to be talking about the interfaces of sleep and anesthesia. And before I give you an outline of the talk, I just want to address an important question. Why should you care? Why should we care as a field about the interfaces of sleep and anesthesia? And I think there are 3 answers to that question. First of all, the interfaces of sleep and anesthesia touch on a fundamental scientific question in the field of anesthesiology, and that is, what is the mechanism of anesthetic-induced unconsciousness? But it's not just a scientific interest, because we know that there are a number of postoperative complications that are related to sleep and sleep disorders. And as you know, as clinicians, and I know, there's an increasing prevalence of patients who have disorders such as obstructive sleep apnea. And finally, we also do a very good job as physicians of depriving and fragmenting sleep. And so many patients in the intensive care unit or on the general medical wards have sleep deprivation or sleep fragmentation. And there are a number of different cognitive outcomes, adverse outcomes associated with that, such as delirium. So I think it is an important topic, both scientifically and clinically. And so in the next 35 or 40 minutes, I'm going to explore 3 questions. First of all, is there a shared phenotype between sleep and anesthesia? How are they the same? How are they different? And is that phenotype driven by shared underlying mechanisms? And I'm going to be focusing on both subcortical and cortical mechanisms of both anesthetic and sleep-induced unconsciousness. And then finally, do those shared mechanisms drive a shared function? How does general anesthesia interact with sleep homeostasis? How do sleep profiles affect anesthetic sensitivity? So first, let me state the obvious, and that is there's no trivial identity relationship between sleep and anesthesia. We know that these are distinct states, and they have distinct traits. And this is something that the public also knows in the past few years in the discussion of the death of Michael Jackson, in which the use of propofol to try to satisfy sleep deprivation came into the public eye, and the question of how sleep and anesthesia were similar or different was also a matter of discussion. So despite the fact that these are distinct states, we still use sleep as a metaphor for general anesthesia. I would venture to guess that most in the audience have done this. I do this. We tell patients, you're going to be going off to sleep now, you're going to be having pleasant dreams. So why do we use this as a metaphor when we know that the 2 states are different? Well, first of all, sleep is really the experiential basis for our knowledge of unconsciousness, and in particular, reversible unconsciousness. So even if the patient presenting to the operating room has no idea what general anesthesia is, they certainly know what it's like to go to sleep and then to wake up again. So they have an intuitive or experiential knowledge of reversible unconsciousness. And furthermore, going to sleep has very positive connotations. It's associated with health, it's associated with a sense of restoration. So it's far more reassuring to tell a patient, you're going to be going to sleep now, you're going to be having pleasant dreams, as opposed to something like, we're going to be inducing pharmacologic coma, but don't worry, it's you'll usually wake up. So we tend not to say things like that. So I can't tell you when this metaphor was first used, but I can tell you that it goes back a long way. And if you look in the first book of the Bible, in Genesis, you'll see the quotation, "And the Lord God caused a deep sleep to fall upon Adam, and he slept, and he took one of his ribs and closed up the flesh in its place." Now, independently of whether your belief system is rooted in the Judeo-Christian tradition, I think you can agree that the concept of surgical anesthesia as a kind of deep sleep is an ancient one. I should also note that God, in his infinite wisdom, had the good taste to become an anesthesiologist before he was a thoracic surgeon. So we use sleep as a metaphor. Why? Well, likely because there's a shared phenotype. There's something about general anesthesia that looks like sleep. But again, these are distinct states, and they have distinct traits. And some of the critically distinct traits are the fact that anesthesia is pharmacologic, whereas sleep is something that's endogenous and spontaneous. Anesthesia is irreversible with respect or reference to a noxious stimulus, whereas sleep is readily reversible. We don't need to put a scalpel on somebody's belly to get them to wake up. We can call their name. And finally, sleep is homeostatic. We need sleep. I don't think anybody would make the claim that we need a general anesthetic. But despite these differences, there are some shared traits, and these traits are almost canonical for defining a state of general anesthesia: hypnosis or unconsciousness, analgesia, amnesia, and finally immobility. And that applies in particular to REM sleep, where, as you know, we have paralysis that occurs. So if we extend this concept of phenotype to a neurophysiological level, you'll also see a number of similarities. And I put up a slide here showing you some work actually by one of my senior colleagues at the University of Michigan, Ralph Leidick. And you're just looking at compressed EEG tracing of a waking state, non-REM or slow-wave sleep, and then something that is characteristic of non-REM, which is called a sleep spindle. If you look at the lower part of the panel, you also see a very similar kind of slow-wave morphology that happens during an inhaled anesthetic. And furthermore, you will also see the presence of spindles. Now, they're not exactly like sleep spindles, but qualitatively you can see that they're similar. And it's also been identified in propofol, or during a propofol anesthetic, during dexmedetomidine. So there's a shared neurophysiologic or electrophysiologic phenotype as well. Similarly, there have been formal comparisons of slow waves during both sleep and propofol-induced unconsciousness. This was published several years ago, and there are a number of similarities. There are slow waves identified in all unconscious states. They spanned a large number of channels, that is EEG channels. There is a consistent frequency and distribution. And finally, and of interest, they had an anterior origin, and they propagated posteriorly. So a similar traveling wave through the brain with slow waves during both sleep and propofol-induced unconsciousness. So we use sleep as a metaphor for general anesthesia. That's likely driven because there's this shared phenotype. But now we want to ask the question, is this shared phenotype driven by shared underlying mechanisms? I'm going to talk about some of the subcortical structures and cortical processes that are involved in that. So this hypothesis was first formally put forward in the mid-90s, again, by my colleague, Dr. Ralph Leidick. And in an editorial in the British Journal, he stated, "Thus it is highly probable that neuronal mechanisms which have evolved for regulating naturally occurring states are preferentially involved in generating some traits characteristic of anesthetic-induced states." This is now sometimes referred to as the shared circuits hypothesis. So sleep and anesthesia are sharing circuits in the brain. And I should also note, within the context of anesthetic mechanisms, this is a shift from thinking about molecular actions to more a systems neuroscience level by exploring a sleep network. So to appreciate this, we need to understand a little bit about the neurobiology of sleep-wake control. And on the left side, you can see a number of different arousal centers. And I talked about this briefly in my talk yesterday on awareness. but a number of different nuclei with different neurotransmitters that are responsible for arousing the cortex. So for example, in the pons, you have the locus ceruleus. It transmits norepinephrine. You also have the laterodorsal and pedunculopontine nuclei. They transmit acetylcholine through the thalamus. You have the dorsal raphe, serotonergic. In the midbrain, you have the ventral tegmental area, which is dopaminergic. And then moving up to the hypothalamus, you have the tumoral mammillary nucleus, which transmits histamine, and you also have orexinergic neurons. So these nuclei have widespread projections to the cortex, and they serve to keep the cortex awake or aroused. But of course, we don't spend all of our time being awake. So as the day moves on, as circadian cues start to shift, as you start to build up a homeostatic pressure for sleep, there's a shift or a flip-flop that goes on in the brain. And now there's another center or set of centers that starts to turn on. And most notably is the ventrolateral preoptic nucleus. This is found in the hypothalamus, and this turns on when you go to sleep. So it's a sleep-active set of neurons. And what it does is, in turn, it starts to inhibit all of those arousal centers. So you have this reciprocal inhibition and excitation between the nuclei that are responsible for arousal and the nuclei that are responsible for sleep. So you can imagine a structure that turns on and puts your brain to sleep would be an attractive candidate for thinking about anesthetic mechanism. And indeed, about 10 years ago, there was a paper in Nature Neuroscience by Mervyn Mays' group. He's now at the University of California at San Francisco, showing that Vilpo becomes metabolically active during general anesthesia, suggesting that it's turning on. There's something about anesthetics that are turning on that area, which might help facilitate a shutdown in the brain. It was really only recently, however, that it was shown that there was a really direct effect on the neurons causing excitation by isoflurane. And I'm going to not get into that so much, but show you what happens when you lesion the Vilpo. There's both an acute and a chronic effect. So this is basically just a dose-response curve. This is in rodents. You have the fraction obtunded and the percentage of isoflurane. So in black is your control, normal dose-response curve with a sham surgery. So that's the control condition. So after Vilpo is lesioned, acutely, or not really acutely, but 6 days afterwards, what you see is a rightward shift in that dose-response curve. So that means there's a relative resistance to the effects of isoflurane, and you need to use more isoflurane to get the same fraction of animals obtunded. So by losing the Vilpo, the animals have become resistant to the effects of isoflurane, which suggests that Vilpo is important in mediating the effects of isoflurane. Now, after that point, things change a bit. The animals cannot sleep. They develop a chronic insomnia. So at 24 days out, you now have a leftward shift. So this homeostatic pressure for sleep has become so profound that the animal actually becomes hypersensitive to the effects of anesthetics. And you have a leftward shift, and you need far less isoflurane to get the same fraction of animals obtunded. So this really speaks to two mechanisms. One would be the anesthetic working through Vilpo, but the other is all these other centers and the drives to sleep affecting anesthetic sensitivity, and I'll talk about that later. So now moving from the subcortical region to the thalamocortical or corticocortical networks, I want to talk about similarities. And for those of you who were here a couple days ago, you saw this experimental setup of the healthy volunteer who has the high-density EEG on and who's getting a burst of transcranial magnetic stimulation. I showed you data from midazolam. Now I'll show you data from sleep. And this was the original article published in 2005 in Science. So here's the healthy volunteer in the waking state. This is really just the evoked potentials, and you can see it moving out greater than 250 milliseconds, which is a time frame that roughly correlates with what is required for conscious processing. Below that, you see the shifting voltage contour map, but really the key is this this line right here, where you see the activation that starts to spread through the brain as different areas of the brain become communicative with one another. And this is something, as I mentioned before in the talk a few days ago, that seems to be characteristic of conscious processing. Now, by contrast, when the healthy volunteers are in a state of non-REM or slow-wave sleep, you still see that stimulation, but it doesn't really go anywhere. So the brain still receives it, but you no longer have these different areas talking to one another. The brain starts to become very modular. As I showed you a few days ago, something very similar happens with general anesthesia. So this is the same experimental setup, and you can see with midazolam-induced unconsciousness that there's a similar limitation, both in space and in time, of the effects of this stimulation. So at both the subcortical and the cortical level, there are definite similarities between sleep mechanisms and anesthetic mechanisms. Now, just because you have a shared trait, I was talking about unconsciousness, doesn't necessarily mean that you have a shared mechanism. So as I noted before, immobility is something that is shared by both anesthesia and sleep, but they have very different mechanisms. As we know from the work in the 1990s by Antonini and also by Rampel, immobility during anesthesia is mediated primarily at the level of the spinal cord. So there's spinal mechanisms. During sleep, however, immobility during REM is generated by supraspinal mechanisms in the brainstem. So just because you have a shared state does not mean that you have shared underlying mechanisms. So we've talked about anesthesia and sleep being a metaphor for anesthesia, likely because of that shared phenotype. And I've just shown you that that shared phenotype is likely driven by shared underlying mechanisms, both at the subcortical and the cortical level. So now this leads to the question of whether there's a shared function between sleep and anesthesia. Can a sleep profile affect anesthetic requirements or anesthetic behavior? And what are the effects of anesthesia on regulating sleep architecture? And so first I'll explore the question of how a sleep profile can affect anesthetic sensitivity. This was a study done in rodents by Avery Tung at the University of Chicago. And basically, these animals were deprived of REM sleep for 24 hours. So leading up to this point, 24 hours of total sleep deprivation. And then the animals were induced with either propofol or isoflurane. Now you see this x-axis is the time to loss of righting reflex. The animals like to be on all fours. So if you put them on their back, they will right themselves and get back on all fours. So in experimental anesthesiology, we use the loss of that reflex as a surrogate for unconsciousness. So this will come up a few times. But you can see that the sleep-deprived group in black had a shorter time to anesthetic induction for both propofol and for isoflurane, suggesting that that need for sleep was somehow satisfied and potentiated the effects of anesthetics. On the other end of the experience, there was a delayed time to emergence in the animals that were sleep deprived. So here's that group in black, and you can see an even more profound effect of delaying recovery after total sleep deprivation. Now, another interesting experiment to address the question of whether a sleep profile or sleep characteristics can affect anesthetic sensitivity happened not in rodents, but in fruit flies. So Drosophila melanogaster is something that's used frequently, and the reason why it's used experimentally is because it's very easy to manipulate from a genetic perspective. So you can generate a number of different mutants and test gene function. So in this case, one mutant that was generated is referred to as the Shaker mini-sleep mutant. It's called Shaker because it's a mutation of a voltage-gated potassium channel that's referred to as Shaker. And these Drosophila actually seem to sleep less. So it's referred to as the mini-sleep mutant. So, well, what does it mean for a fruit fly to sleep? Essentially, it's really a measure of quiescence versus activity. And so the surrogate here is the Drosophila not flying around. So what these investigators did is they tested the wild-type phenotype, so the normal fruit fly, and compared it to this mini-sleep mutant that seems to have less of a need for sleep. And they exposed them to isoflurane. And you're looking at a dose-response curve. So this is the wild-type curve. So these are the Drosophila with a normal genetic background. And this is the Shaker mutant. And what you can see is that there's a rightward shift in this dose-response curve. So these Shaker mutants, the ones that don't need much sleep, seem to have a resistance to the effects of the anesthetic. It might be like somebody who's coming into the operating room who has had amphetamines and who has less sleep need but has an increased need for the anesthetic. But the difference in this example is that this is actually under genetic control. So you have a single gene that is not only controlling the complex behavior of sleep, but it's also controlling sensitivity to general anesthetics. So now let's explore the other side of this relationship and see how anesthesia can affect sleep profiles. So there are three general options that we could explore. First of all, anesthesia could satisfy sleep debt, much in the way that sleep does itself. Anesthesia could, like the waking state, lead to the accrual of sleep debt, or anesthesia could be neutral. It's neither accruing nor dissipating sleep debt. So to explore this question, again, Avery Tung's group, using rodents, sleep-deprived animals for 24 hours, then he let them either sleep or they were exposed to 6 hours of propofol. So just to help you with this graph, the animals are deprived for 24 hours up to this point. One group is receiving intralipid, and now we're looking at non-REM sleep. And the other group was receiving propofol during this period. So then the key is, well, how do they look afterwards? How does that sleep recovery versus propofol recovery look? And in terms of non-REM sleep, there was really no difference between the propofol and the intralipid group. Now you see these markers of statistical significance. That's a difference from the baseline condition that had no sleep deprivation. But the propofol versus intralipid had no differences. Even more surprising, there was a similar case for REM sleep. So these animals were deprived, and there were no differences in REM rebound. What I mean by rebound is if you're sleep deprived or if you're REM deprived, and then you're allowed to sleep, you're going to have an overdrive to make up for that. And that's referred to as a rebound phenomenon. So this is even more surprising because I've shown you that anesthetics or a general anesthetic can kind of look like slow wave sleep, but there's really nothing in a general anesthetic that looks like REM sleep. REM sleep on an EEG looks like a waking brain, which is why it used to be referred to as paradoxical sleep, because the cortex is active and yet you're even more disconnected from what's going on in the environment. So to answer the question of the relationship of anesthesia and sleep homeostasis, propofol seems to fit into the first category. That is, it appears to satisfy sleep debt, both non-REM and REM sleep. So a few years back, I asked the question, well, what about inhaled anesthetics? This is something that we're using probably more commonly. It has a more diverse mechanism of action than propofol. Propofol more specific for the GABA-A receptor. And as I just mentioned, it was not at all obvious to me that a general anesthetic should satisfy a REM sleep-like state, which is really a more activated state of the cortex. So I'm going to talk about two studies that we performed. The first was using isoflurane in rodents with selective REM sleep deprivation. So how do you selectively deprive an animal of REM sleep? It's a very low-tech methodology. You basically take a big basin, you put water in it, you take flower pots and you turn them upside down and you put them in the water, and then you put the animal on top of the flower pot. So the animal's sitting there on the flower pot like this. It goes to sleep. It goes into non-REM sleep, and it can stay on the flower pot because it is retaining muscle tone. When the animal goes into REM sleep, it loses muscle tone, it falls off the flower pot into the water, it wakes up, and then it scrambles to get back on the flower pot because they hate being in that water. So it's very low tech. It was actually developed in the 1960s by Jouvet in France. So this is the technique we use to selectively deprive REM sleep, and there are basically four conditions. The first two had, well, actually condition 1 and 3, the animals are doing what they want, ad lib, sleep, or wake states. Conditions 2 or 4, we had selective REM sleep deprivation. Then the animals were either allowed to recover naturally or they got isoflurane. So really what we're comparing here is, what did this isoflurane do after REM-DEP compared to this ad-lib sleep state after REM-DEP? We were also interested in what was going on in the hippocampus, and this is a picture of a hippocampal slice. And the reason for that is that the hippocampus generates a rhythm called theta, which has a characteristic sawtooth pattern, which is prominent in one of the markers of an animal being in REM sleep. And we wanted to see what was going on in terms of those hippocampal theta dynamics during sleep and anesthesia. And here's a picture of the animals. They have implanted EEG. They've been induced in a chamber with isoflurane after REM sleep deprivation, and then they basically breathe spontaneously through a nose cone. We have a little monitoring device that's similar to a pulse ox. It's actually called a mouse ox, and it gives you an oximetry value as well as some other surrogates of heart rate and blood pressure. And importantly, isoflurane was titrated to unresponsiveness without burst suppression, and that will become significant later. So here's what we saw. This is no anesthetic involved up here. So the animals are, ones in black were REM sleep deprived for 24 hours up to this point. The ones in gray got to do what they want for the 24 hours up to this point. And now we turn them loose and we let them do what they want. And what you can see is that the animals that are REM deprived in black have this big increase in REM sleep. That's REM rebound and is to be expected. Now that REM debt gets discharged within the first 4 hours, because you can see the subsequent 4 hours, they're not different between the non-deprived and the deprived group. So now let's look at the situation with isoflurane. So the group in black had REM sleep deprivation up to this point. The group in gray got to do what they wanted up until this point, and then they both have isoflurane. Now, if isoflurane satisfied the need for REM sleep as well as natural sleep did, we would expect that these 4 hours would look something like this. In other words, we would expect that the REM sleep debt would have dissipated and discharged during this period. But it didn't. And you can tell pretty clearly that the rebound going on here looks very similar to the rebound going on here. So in a sense, there was really just a circadian shift. And it's pretty remarkable when you think about it. The brain, despite this full-on anesthetic, still retained a memory for how much REM sleep it needed. And so when the animal woke up, it started discharging that. The brain just picked up where it left off. It knew exactly what it needed, and it discharged that REM sleep debt in 4 hours. Now, I pointed out this study. It was actually performed in part in New Zealand because I mentioned this circadian shift. This was a study of bees, actually, showing that, in a sense, anesthetics induce a kind of jet lag. They lead to circadian shifts, and it was a very clever set of experiments. I just wanted to point that out for your interest. So I'm not going to get into all of the work that we did with the hippocampal theta, but just to point out that that sleep deprivation, and this is during an anesthetic, not sleep, really didn't seem to have any effect on the expression of theta. And what that suggests is that the theta that is generated during anesthesia, and there is theta generated, is probably due to different mechanisms. And we're starting to probe that in even more detail, and I'm not going to go into the study. We've just submitted it to the Journal of Neuroscience. But these animals actually had electrodes placed in neurons in the hippocampus, and there's something called a place cell. So if a rat is running on a maze and it gets to a certain point, there's going to be a certain neuron that starts to chatter that you'll hear. When it moves away, it will stop chattering. When it gets back there, it will start chattering again. So there's this map of space that happens in the hippocampus. And we've been exploring what happens during the waking state, after sleep, and during isoflurane. And you can see this sort of smearing effect of the spatial map after isoflurane. So that's just something to point out in terms of what direction we're going, in terms of getting into more details. So, to answer the question about anesthesia and sleep homeostasis with isoflurane, isoflurane really fit into the third category, at least with respect to REM sleep. It appeared to be neutral with respect to sleep debt. It didn't discharge the REM sleep debt, and it didn't necessarily increase it. So it was neutral, it was suspended, and the brain remembered how much REM sleep it needed. So we moved on to a second study. We wanted to see, well, was this just specific to isoflurane, or is it more generally true with the halogenated ethers, number one. Number two, we had just looked at REM sleep deprivation, and we wanted to look at total sleep deprivation. So here's the experimental setup. We had 36 hours of recording. These were very long experiments and sometimes kept us there late into the night. So this condition had 12 hours of total sleep deprivation during the light stage of the day, which is when the animals like to sleep. Remember that these rodents are nocturnal. And then we let them recover however they wanted, or they got sleep deprivation for 12 hours, and that was followed by 6 hours of a sevoflurane anesthetic. Now, in this situation, we actually titrated the sevoflurane to burst suppression. So why did we do that, or why is that important? Well, if you think about these other studies that have been published in the literature, they didn't really titrate the anesthetic to an anesthetic-specific endpoint. So, for example, we're seeing slow waves while we're delivering the anesthesia, but how do we know that's not sleep? How do we know that the animal is not sort of going into a sleep state and recovering sleep? And so what we wanted to do is we wanted to titrate the sevoflurane to an endpoint that we knew was specific for an anesthetic. You do not get burst suppression during natural sleep. You go into slow wave, then you go into REM. When you have an anesthetic, you have slow waves, you give more anesthetic, ultimately you have burst suppression, and you can see that characteristic pattern of quiescence followed by the burst of activity. So we knew that the effects that we were seeing were going to be attributable to the anesthetic and not to just slow wave homology or slow wave sleep itself. So the animals were deprived, or one group is deprived up until this point, and again we want to see what happens afterwards when we allow them to sleep or get sevoflurane. So let me just take you through this. This is for non-REM sleep. The red bar is the group that had no sleep deprivation. So here's their non-REM sleep here. You can see this is later in the day, and at night, again, the animals are nocturnal, so they're going to be more awake during this time. And then during the light phase, that REM sleep goes up, because that's when they like to sleep. The blue group had the sleep deprivation. They were allowed to sleep here, and you can see that significant increase. So that's a non-REM rebound phenomenon. And then that gets discharged within these 12 hours, and then it goes back to a baseline. The third group, instead of sleeping here, got 6 hours of sevoflurane. Well, now let's look what happened afterwards. When it got out, this significance, or this significant difference, was with respect to the deprived group, but there was no difference with respect to the non-deprived group. And you can see there's no rebound over here. So what does that mean? What it means is that it took the animals 12 hours of natural sleep-wake cycles to satisfy that need for sleep after deprivation. But the animals that had a 6-hour deep sevoflurane anesthetic, they came out of it recovered. So they did not show any signs of being sleep deprived after the anesthetic was done. And that's supported also by another marker of sleep restoration, which is delta power, delta being the slow waves, 0.5 to 4 hertz. You can see the animals that were sleep deprived, they have an increased delta power, which is what you'd expect. It's a more intense sleep, intense slow wave sleep. And then there's this slight negative rebound. And then you can see with the sevoflurane group, the green bars are significantly lower. So showing a lower delta power is essentially a marker for the animal needing less sleep. So basically, that sevoflurane anesthetic was a kind of super sleep. It only took 6 hours to satisfy that need versus 12 hours of normal sleep cycles. But this wasn't true for REM sleep. So again, the red group is the group that got to do whatever it wanted. You see it's low during the day. REM sleep starts to go up at, actually REM sleep starts to go up during the day, and it's low at night. The sleep deprivation group, you get this big REM rebound, these big blue bars coming up over here, and it took 12 hours to discharge that debt. Now what happened with the sevoflurane group, when they came out, they also had REM rebound, and that REM rebound continued into the next time epoch. So like the isoflurane, the animals came out, they knew they had 12 hours of REM sleep to make up, and then they made it up. So this is showing a dissociation between non-REM or slow wave sleep homeostasis and REM sleep homeostasis that's going on during the anesthetic. So there have been a number of converging findings in the field. It's been shown that isoflurane and desflurane both satisfy slow-wave sleep homeostasis after deprivation, but sevoflurane and desflurane result in a selective REM sleep rebound. So the picture that Avery Tung's group gave us in terms of how sleep and anesthesia related to one another was very balanced. If you had sleep deprivation, you gave propofol, you'd see there was an effect there. If you gave propofol, it also satisfied all these components of sleep. So it was this balanced picture. What we've shown is a different paradigm for the halogenated ethers, which is that there's a profound effect on non-REM sleep. It cuts down the recovery time by half, but there's a block of REM sleep discharge. So we're dissociating these two homeostatic processes. So what might that mean for us clinically? Now keep in mind that the experiments that I've talked about are in animals, and number two, they're only looking at anesthesia. We're not doing surgery, we're not giving opioids, we're not stressing the animals. So the perioperative picture is much more complex. But let's think about just the anesthetic effects. You might think of a patient coming in a certain behavioral state or a certain brain state who gets propofol or sevoflurane and kind of comes out the other end in a similar state in terms of the sleep profile, the anesthetic wears off, the patient gets right back to where they were. But imagine a patient coming in with obstructive sleep apnea, or somebody coming from an ICU who has sleep deprivation or sleep fragmentation. If they come to you deprived and they get propofol, based on the preclinical data, they may potentially have a satisfaction of both non-REM and REM sleep homeostasis. By contrast, the same patient who is getting sevoflurane, based on these preclinical data, would come out in a REM-deprived state. So they would have the non-REM sleep satisfied during the sevoflurane anesthetic, but the brain would still be recording, still be generating REM sleep debt. This actually might have clinical consequences. If you have a patient who has obstructive sleep apnea, when they go into REM, and especially if it's a rebound recovery REM, they're paralyzed, as we all are during REM sleep, but they also start to lose muscle tone, genioglossus tone, that helps keep the airway patent. So there might actually be consequences in terms of patients who have sleep apnea and hypoxic episodes, and this is something that can actually happen days out. If you have opioids or excessive opioids, you can suppress REM sleep. It starts to rebound at day 2 or day 3, and that's where bad things can happen. I also wonder what the effects are in terms of cognition. If you have REM sleep intrusions or if you're disrupting normal cognition, could this be associated with delirium? So thinking about how sleep restores our cognitive powers and our cognitive functions, and how anesthetics may or may not do that could have implications for post-anesthesia care or for sedation in the ICU. So to summarize, sleep and anesthesia are distinct states, they have distinct traits, but they share a number of common features, some of which are canonical for the definition of a successful general anesthetic. Sleep and anesthesia have overlapping mechanisms, both at the subcortical and the thalamocortical level, and the functional relationship of sleep and anesthesia is bidirectional. So a sleep profile can affect anesthetic sensitivity, and anesthetics can alter sleep architecture. Intravenous and inhaled anesthetics have distinct effects on sleep homeostasis. This may have clinical significance. It's yet to be tested, but I think it's something that is testable. And even though there's a connection between sleep and anesthesia, the data I've shown you suggests that there's no indiscriminate relationship. The relationship is likely state-specific. Are you talking about non-REM or REM? And it's likely drug-specific. Are you talking about propofol, or are you talking about one of the halogenated ethers? So with that, as I did a few days ago, I'd like to thank my research team and my funding sources, and acknowledge the Society for Neuroscience and Anesthesiology and Critical Care. And before I was a physician, I studied philosophy, and I wanted to conclude with a quotation from Nietzsche, who is a philosopher who liked to turn values on their head. And he said, "Sleeping is no mean art. For its sake, we must stay awake all day." And I thank those of you who managed to stay awake during this talk, and I look forward to speaking with you later. Thank you.